Good evening, everybody in the Americas. Good late evening to those of you in Europe, and a bright good morning to those of you in the Asia Pacific region. This is our third update call for the OHDSI COVID Study-A-Thon. I'm going to walk you through the remarkable progress we've been making during our first day. So many of you joined us for our Study-A-Thon kickoff, which was only a short 16 hours ago. It was 3 a.m. for me, and I enjoyed the fact that we were able to start off with hearing a great presentation from Danny, from George, and from Peter, outlining the real problem that we're facing and why we as the OHDSI community should be able to tackle this together through collaboration. Craig Saxon's done a great job of actually posting the video of this. So if you're in the MS Teams, you can actually get to the video on the general channel. And if you're watching this externally, the video has been posted on YouTube, and you can get to it at odyssey.org/COVID-19 updates. Now, one of the things that Danny and Peter and George and I boldly proclaimed was there's this big health problem that we know that's going on in the world, and that if we could just collaborate together, we'd be able to make remarkable progress. But I'll let you in on a little bit of a secret. We didn't know how this was going to go. We've never done a virtual study-a-thon. We sure as heck haven't done a virtual study-a-thon with several hundred individuals trying to get together. So we were going to kind of figure this out along the way. So I'm really excited to be able to share with you the progress we've made on day one, just 16 hours into this journey together. First, I think we need to start with the bad news. COVID-19 is still a problem in the world. We've crossed some pretty unfortunate milestones. There's now over 500 million patients who have been inflicted by this disease, over 23,000 patients who have lost their lives to this. Unfortunately, the work we're going to do today and tomorrow isn't going to make this problem go away, but it should provide us firm motivation for why we're all here together and the difference that we can make to try to generate reliable evidence that's going to hope to stem this tide of this pandemic. It's been particularly sobering for me to hear the news of my colleagues who are clinicians and are on the front lines right now, who are really struggling with terrible conditions and healthcare systems that are just not equipped to take care of the patients that are being faced. And so I do want to just take a moment to acknowledge everybody who's in a hardship right now. Thank those first responders and healthcare professionals who are working hard, and to wish all those patients that are inflicted by this disease a speedy recovery. Now, we said 16 hours ago that we wanted to be able to collaborate together to try to generate some real-world evidence to support various problems that we thought if we could generate real-world evidence across our community, we'd be able to inform the ongoing decisions that need to be made by policymakers, healthcare professionals, and patients. And I showed you this slide early this morning that specifically outlined that we had a series of characterization studies that we wanted to tackle, a series of prediction studies, and a series of estimation studies. We described how we were going to characterize patients who are COVID positive, and we were going to benchmark them against patients who have influenza signs and symptoms to be able to provide a background context. We also talked about the need to understand invasive treatments for respiratory distress. In the prediction work, we talked about the need for having the channel that's being led by Peter and Jenna and Ross focused on questions about trying to help flatten this curve by asking questions about which patients are actually most at risk of requiring hospitalization, which patients hospitalized are going to require more intensive services. And then on the estimation front, there's a lot of speculative diseases, speculative treatments that are being considered either as prophylaxis or treatment of this disease. Those include hydroxychloroquine, IL-6 inhibitors, HIV and Hep C protease inhibitors. And these treatments, we said that we would try to design studies to actually answer questions of causal inference about the safety and the effectiveness of these products on various viral diseases. We also wanted to actually explore the interaction between the coronavirus in the ACE2 pathway, and particularly its effect on ACE inhibitors and angiotensin receptor blockers, recognizing that a large proportion of patients with hypertension may need this information. Now, we described that for every study that we were going to do, we were going to need to march through various competencies. We were going to need to conduct literature review. We're going to need to develop and evaluate phenotypes. We were going to need to develop study packages. We were going to execute those studies across our network, and we were going to need to perform clinical review and dissemination of the results. Now, this was the picture that we laid out, and Peter boldly said, we're going to go from to be done to completed by the end of the 4 days. And while I certainly share his enthusiasm and desire that that's where we want to go, the truth of the matter is we don't know how far we're going to get on this journey. What I'm remarkably impressed and proud of everybody on this community and everybody's work is how much progress we've made in a few short hours. Just 16 hours ago, we laid out this idea and challenged you all to join the community and try to make whatever contributions you can make across any of the cells in this space. And here's where we are right now. As of 7 p.m. Eastern Time on day 1, we are already actively in progress in conducting the literature review for our characterization studies. We've nearly completed the literature review for our prediction and estimation studies based on the great work of a lot of individuals. The phenotype development work is hard at work trying to develop phenotypes that can be used across our characterization, estimation, and prediction studies. And I'll spend a little bit more time on that. And already right now, we have packages that are being developed and on their way on github.com/Odyssey and Odyssey Studies to show the progress that we're making. Not only that, but the community's come together to start to posit new questions that we want to tackle. And already there's been consensus within the characterization group that a new channel will be formed focused not only on patients who are actually diagnosed with COVID-19, but those who are just simply being tested for that. And recognizing that's an important public health question, and I'm excited to see a new effort spin up to try to explore that space. So to look at where we were just 16 short hours ago to where we are now with all of the tremendous progress going in full swing across all of our studies, I just want to give everybody a congratulations for all the hard work that you're putting into this. Now, what I'd like to do quickly is just provide a quick summary highlights through all of the channels and the progress that has been made. And so to do that, I'm going to switch my screen here to show the channels live. So Jenny and Nige have been hard at work on our literature review and protocol development group. There's been tremendous progress actually being made in terms of producing finalized protocols. I want to specifically highlight that it's been great to see how many people have raised their hand and suggested opportunities for them to jump in and contribute to the review. And it's exciting to see the progress being made in terms of finishing some of these literature reviews. All of this information coming from the literature reviews is directly feeding into developing our background sections for all of our study reports and manuscripts. It is directly informing the designs of our studies. I'm going to skip phenotype development and come back to that in a second. The study package development group, what's really exciting here to see the group being led by Martijn. Martijn has actually released a package called COVID-19 cohort evaluation to allow us to begin characterizing cohorts. And we've also started to prepare packages to actually execute the population level effect estimation studies. And with the hard work of Mark and Jamie Weaver and others, we're making good progress in trying to package up a study that can conduct both self-controlled case series and comparative cohort analyses across our various clinical hypotheses. And the other competency is our study execution. I just want to give a shout out to Kristen and Talita. They've been doing a great job of bringing together all of our data partners and preparing them for the ability to execute studies. And it's really exciting to see how many folks who have actually joined us even in the last 16 hours, who've heard about what we're doing in the Study-A-Thon, recognize that they have access to data, and are eagerly anticipating executing some of these packages that are running across the community. So I want to thank all of you who have data and are contributing. Exciting to see data from Korea, from Spain, from Maine, from Colombia, everybody pitching in and being prepared to execute packages as soon as they're available. Now, if I go through the studies on a characterization front, Ed and Chan have made tremendous progress in defining and getting the group to coalesce around some specific questions that we want to focus on. And it's really valuable for us to see that the group has seemed to coalesce around the idea that there's a need to characterize COVID positive patients, but there's also a new need to explore patients who are being tested for coronavirus. And that's going to stimulate a new work group that's going to be focused on that over the next day. So great job on the characterization front. On the prediction front, there's been a lot of progress in critical examination of the clinical problems of how can we apply prediction to flatten the curve by looking at which patients are at risk of hospitalization and which patients that are hospitalized are at risk for intensive services. There's been really thoughtful conversations specifically going on around how exactly are we going to apply prediction models in a clinical context. And while that thoughtful deliberation's been going on, in parallel, this group has made tremendous progress in actually designing out a prediction package and starting to prepare that to be made available for the community based on cohorts that the phenotype development group has been actively working on. So great job, prediction group. On the estimation front, I'll start here with the hydroxychloroquine study. There's been tremendous progress in bringing together. It turns out that with the hydroxychloroquine, there's a wealth of literature about the safety of this product in RA, and there's a lot of speculative information going on about whether or not hydroxychloroquine is going to work in COVID-19. And the group's done a great job of trying to summarize that information and reaching the point where we've got a nice drafted protocol in place that our colleagues in Europe are looking to finalize by the morning before I wake up. On the IL-6 and JAK inhibitors, the team has also made progress. Danny's posted version 1.1 of the study protocol for the entire team to review. So we're already seeing a protocol in place that's laying out the comparisons that are going to be made and how we can draw insights about whether or not immunosuppressants seem to have a potential valuable effect in viral diseases. Toledo and Albert have made tremendous progress on the Hep C and HIV protease inhibitor work. In particular, what was very clever of them is they recognized that a lot of the work that they were doing was largely consistent between these work groups. And so these two groups have been working together to try to develop a common protocol that can simply branch off based on the exposures. So thank you, Albert and Toledo, for all your hard work. On the ACE inhibitor front, really nice progress being made. Mitch has done a really nice job of summarizing the literature and providing a context of what we know. It's pretty remarkable about how much speculation is going on versus what we know from hard data. And it's pretty obvious to me that this is an area that we can make direct contributions to this weekend, but also we're going to be able to design very robust studies that allow us to more thoroughly investigate these questions about ACE inhibitors as COVID data accumulates. The group has been making progress on thinking about the particular study designs that they're focused on. So great job to Mitch, Chan, and the entire team in this front. And last but not least, I wanna since I get to run this show, I'm gonna specifically give a big shout out to my phenotype development and evaluation group. We set forth a pretty ridiculously ambitious target at the start. I'll show you the crazy thing that I showed them as to what I wanted them to do. I specifically said, okay, everybody else on those study work groups, they're saying that they've got to do an individual study. But of course, every single study requires lots of phenotypes. And every single phenotype requires us to go through a diligent process of writing a clinical description, performing a literature review, designing a cohort in Atlas, evaluating it, running cohort diagnostics, running Phe evaluator, and ultimately publishing a final cohort. And rather than scaring people off, I've been amazed by how much people have taken that call and said, okay, let's get her done. This is the spreadsheet that we were monitoring our progress on phenotypes today. And this is the progress that we've already made in 16 short hours. Everything you see in orange is actively assigned and in progress by the community. Everything in green is already done. That's right, we've got literature reviews done, we've got initial cohort designs done across a large handful of phenotypes in just under 1 day. Now, I'm showing you that and it's really hard to read, but I want to toggle back to the teams to show you exactly what that looks like. In our phenotype tracker, I'm gonna, let's see, make that big so you can see it. This is not just some abstract artwork. You can actually drill down into every phenotype that we're investigating, and you can see for every phenotype who is taking the action to write that clinical description. Where can you find that clinical description? You can see who's performing the literature review and where you can find the abstraction of the information. And you can follow this path forward. I have to say I'm really honored and impressed by how much people have taken this on, that this is clearly something that no one small group or individual could take on. But now that we've got an army of collaborators working together, we're gonna be able to knock this out of the park in terms of producing phenotypes and rapidly evaluate them. Also want to give shout out to Gautam and Aza and Jill and Rupa, who are actively preparing to be able to rapidly run cohort diagnostics on all of these cohorts the moment they become available. So great job. Now, that sounds all good. That sounds like plans, but some people might be wondering, yeah, that sounds good, but show me real tangible progress. So let's show you some real tangible progress. What I'm showing you here is our Atlas environment that was stood up by Lee Evans and Anthony Senna and James Wiggin. This is atlas-covid19.odyssey.org. This is our environment that is serving as our place to collaborate and design concept sets, cohorts, and analyses together. And if you look at this screen, this was an environment that was blank at the time that we started this morning. And as of right now, 7 p.m. Eastern time on day 1, we already have had 104 cohorts generated by our community. These cohorts are still not finalized, still not done, but it's demonstrating and a testament to all the hard work that's going on. Those cohorts are being built up by the building blocks of concept sets that are being actively worked on. We already have in place characterization analyses within Atlas that summarize some of the cohorts that are in place. And we've been able to use those cohorts to produce speculative estimation designs for the studies that the estimation channels are already working on their protocol, and to design patient-level prediction studies. So the phenotype work group has really done amazing work in preparing all of the core inputs that are going to enable the studies. And it's going to be important for us across all of our channels that as we finalize and iterate on our descriptions of what we want, that we collaborate closely to make sure that we deliver an implementation that's consistent with that. Now, cohort definitions are nice. That's a piece of code that one could run. The question is, what happens when we run that code across the data network? And so I want to give a big shout out to Martijn Schumi, who has already released the COVID-19 cohort evaluation package. And we've been able to post our initial preliminary results of cohort definitions out on data.odyssey.org. Just to summarize one example, I'm showing you here in cohort diagnostics some of the results from our initial efficacy outcomes. Here I'm showing you the definition is episodes of influenza symptoms and complications. We've developed a cohort definition and have run it across data in the US, the UK, and Japan. And here we're able to plot the incidence of that cohort over time, stratified by age and gender. And this has actually provided us a great insight into being able to clean up some of the cohort definitions that require iteration after looking from the data, but also giving us confidence that once we actually get to a point that a cohort definition looks promising, then we're ready to actually use it as an input to all of our subsequent studies. On the efficacy front, we've created cohorts already for influenza symptoms, ECMO procedures, ventilation and intubation. For long-term safety outcomes, we've actually created a range of outcomes leveraging some of the great cohort definitions that had already been established for the LEGEND hypertension study. So now you can explore issues associated with pneumonia and respiratory distress, but also outcomes that we had previously studied in other OHDSI research, including acute renal failure, pancreatitis, heart failure, and cardiovascular diseases. All of these are available for you to explore at data.odyssey.org. So great job to Martijn and the team for getting all of this prepared. So what's the journey ahead? Well, this got to be my kickoff that I've got, and I'll be handing it over to Danny for the 3 a.m. Eastern call. So I just want to drop a big challenge to Danny and say, look what my side of the hemisphere was able to do in the last 8 hours. Let's see your group follow suit with some big time deliveries. The journey ahead in the next 8 hours, I think where we're going to be focused in a lot of our time is focusing on trying to finalize our protocols for all of our studies. On the characterization group, there's been tremendous discussion made, and now it's time for us to really write down the final decisions and reach consensus so that we can hand over those definitions of our populations and features to the phenotype group to build, and so that we can start preparing the study package. And the fact that there was that insight that we want to actually create a new study to examine patients who are tested with COVID-19, that means we need to initiate that work and start a protocol on that. So I hope over the next 8 hours we'll make great progress there. On the prediction front, we've been seeing tremendous discussions going on around finalizing the prediction problem. I think where we're at right now is we want to be able to try to provide some initial phenotypes so we can start the process of iteratively training models across different databases. And so I hope that we'll be able to position to look at some results, certainly within the next couple updates from the prediction group. And then on the estimation front, as you saw across the multiple channels, it looks like the target that's been set is to try to produce some initial protocols that are drafted and finalized by the tomorrow morning review. So I hope that when I get up at 3 a.m. to listen to the update, I'm going to see remarkable progress from our colleagues around the world in trying to move that forward. With that, I'm going to stop my update. We'll take a moment. If there's any questions, you can post your questions on the in this forum in the live event, and I'd be happy to answer them if you have any. And I see that we've got over, actually, I failed to mention, and I apologize. We've got over 50 people attending this meeting, and what's remarkable is we have over 300, I think it's 347 by latest tally is the number of participants in our Study-A-Thon. So I really want to thank everybody for joining in and diving into this. I'm gonna start looking at the questions, but I know Peter, you're on, and you've been starting to answer some of the questions. So maybe if you want to help me out and start answering some of the questions, and I'll start reviewing these.
Yeah, there was one question about whether we actually also have the intention to do other type of studies than the ones that are in the sheet, and I answered to the question that of course, yes. So this is just the start of a longer journey that we're on, and we really invite everyone to suggest other questions that we can work on in the upcoming months. So yeah, if you join the team, you have the opportunity to post your questions in one of the channels that's all about that.
All right. And we had a comment from Pradeep at the CDC asking how you could help, and I think Martijn's replied to you privately with a suggestion there, but there's several areas where you could help. But one of the most notable things I want to make sure that we're focused on, particularly given that you're at the CDC, is if there are questions that you all need as a public health agency that you think we could help, we want to hear those questions and make sure that we're responsive to those. So please use the forums and all of the channels as well as the general forum to share your ideas. And if you have specific ideas of individual questions, go ahead and use our support for analytics and support for new study design questions channel to initiate that conversation. Giannis had a very important question. How can a community be so awesome? It's really hard to imagine. I know it's not because of leadership, but I will say it is because of the community. So thank you for that. Question came in about, are we going to be looking at additional risks of patients with lung and other cancers? I know that, so one area that I didn't see progress when I was doing a quick summary of the review, in the characterization work group, we initially had targeted the idea that we were going to do subgroup analyses, and those subgroup analyses would target specific subpopulations. And at least on the tentative list, there was the idea of patients with COPD and patients with cancer being subpopulations. I believe that the group's been focusing mainly on making sure that we get the overall effects of patients with COVID-19 summarized first, but absolutely, I'd encourage you, if you're thinking about specific subpopulations, please raise that in the characterization channel to get that going. Another comment that has been made was about the ACE analyses and whether or not that is going to be stratified by age and gender. I think if there's anybody on the ACE group that wants to just comment in the chat window, certainly I'd like to hear that. I have not actually seen specific expectations around subgroup analyses. I think the initial focus was going to be main effects for the ACE analyses. However, I will say that what it looks like based on the initial data that we saw from our colleagues in Korea is that we're likely at this point to probably only be able to focus on a initial characterization analysis to understand how much use of ACE inhibitors and ARBs and other antihypertensives are observed in the population. But our goal, remember, is that we're not trying to be done at the end of this study-a-thon. We're trying to actually have this be the start of something. And so designing the study to be appropriate such that we can execute it whenever the data are available is an important task for us to go after. Question, are we behind on anything in your mind? A hard question for me to answer because I think every time we do something crazy here in OHDSI, we always seem to set ridiculously ambitious expectations and then we always seem to exceed them somehow. And it's a real testament to all of your hard work in delivering this. From what I'm seeing, I don't actually think of the target is we have to be all done by the end of Sunday, but rather we have to be making really good progress on all of these questions. What I would like to see us do as a community, though, is make sure that we stay focused on delivering some tangible products each and every cycle, each and every day. So as it relates to that, if you're finding yourself having a hard time focusing because there's so many activities going on all at once, I would just encourage you to try to stay focused on one area and drive that to done. And I think that'll really help. We don't need everybody to do a little bit all over the place. We need everybody to do a little bit in one specific place, and then we can build on each other's work to make that progress go. Next question. Will prediction models of COVID be adjusted for triage? That is where patients are at higher risk are being untreated. So I think this is one of the open questions that the prediction group is actually actively wrestling with. I don't know, Peter, if you want to comment on this, but I know that you guys have been having active deliberations on specifically the decision context. Do you want to talk on that?
Yeah, you're absolutely right. So we're really struggling with how to define what we call the target cohort in the cohort in which we make these decisions. So the nice thing of the package, though, is that we can try multiple target cohorts and can do also sensitivity analysis on those definitions. So it's really an ongoing discussions, and I like the fact that there are many people active in that and trying to figure out what the best context is for our prediction model. I don't think we have made up our mind yet, so it's really ongoing. So I think we can give more updates on that in one of the upcoming calls.
Well, another question was if there's been any progress on the phenotypes for COVID-19. So absolutely, yes. The vocabulary group did a remarkable job of actually preparing concept sets for COVID-19 diagnosis, as well as associated procedures and treatments. So we should actually have those prepared. Actually, I think that'll be probably prepared in just like the next hour or two. An important caveat there is that right now we've been trying to leverage data that's at IQVIA and Janssen to do the initial triage of running cohort diagnostics and p-evaluator. Since none of us have data that contain COVID-19, we're going to be needing to rely on other members of the community, including our colleagues in Korea and Colombia who have data that has COVID-19 to test those cohort definitions for us. But so long as people have followed the vocabulary guidance that was prepared by Christian and the team and posted on the wiki, we should be in good shape. The good thing about having a new disease is there isn't lots of different ways to screw it up. There's only one standard concept that needs to be used, and hopefully we're making sure that we're all aligned to that. Any other questions? All right. If not, I want to thank you all. It's been a remarkable first 16 hours of this journey together. I want to thank those of you who are staying up late in Europe and extremely late in Asia, or maybe you're getting up early. I want to thank you for participating. For those of you who are watching this recording, keep up the hard work and jump in on this. I look forward, so I'm handing off the baton here to Danny, who will be leading you on our next update in 8 hours. And I look forward to seeing all the remarkable progress that all of you make from now until then. Thank you all very much. I'll see you on the channels.